Thanks very much for having me here. So I appreciate it. It's nice to be in Budapest. I've been here once before. It's changed a lot. I was here in 1989, interrailing as a young medical student, younger than all of you are now, but before a lot of you were born, I guess. And it was fantastic. I really enjoyed Budapest, and it's been too long since I've been back, so it's great to be back. So I realize this is a mixed audience. Not everybody's from dermatology, but so I wasn't really quite sure what subject to pick. But I just thought I might take a kind of a half a century view on how we've developed new therapies. You know, the whole community around dermatology have developed that, and a little bit of forward looking as well. And then we'll take some questions. But I just thought it might be an interesting thing that maybe there'd be something of general interest to people from different backgrounds. So we're going fine here, yeah. So I wanted to look a little bit about how drugs have been developed for dermatology patients. These radical therapeutic changes over the last half century, developments in immune-mediated inflammatory diseases a little bit more broadly maybe, a little bit about rare diseases, and what the future might look for both of these conditions and where we might be traveling with them. And it's been inspirational to see all of you so early in your careers engaging with the science and the evidence, and hopefully this can be a little bit of inspiration for some of you. So there's two ways to get to patients with skin disease, get drugs. One is through new development, where somebody says, let's develop a drug for a dermatological reason. I can tell you that's relatively rare in the history of drug development, and I'll show you why. And the other is repurposing of already approved drugs for new unapproved indications, and then sometimes taking them through clinical development programs as well. So what's drug repurposing? Because this is, it's the identification of new uses for existing or investigational drugs. And most drugs that are used in dermatology are repurposed from their original targets. And I'm going to show you some examples. We are probably the leading specialty for repurposing drugs. We're like magpies, you know, those little birds that just pick anything that looks shiny and see can we use it for our patients. And it's something that makes dermatology really very interesting in many ways. So just to go back in a little bit of history, so in 1949, Kendall, who subsequently got a Nobel Prize, isolated what he called substance F from adrenal glands. In the first half of the last century, people were mashing up livers and adrenal glands and getting active molecules out of them. And that was where hydrocortisone or cortisol later got named, but it's called substance F immediately. So 1949, that was isolated and very anti-inflammatory. And then really 3 years later, this probably the most consequential dermatologist of the last century, Marion Sulzberger, working in New York, went to Merck, who are still around, said, what about this substance F? Could we make it into a cream? We heard somebody talk about topical JAK inhibitors, where it's a ruxolitinib, originally approved for a bone marrow cancer, JAK2 positive mutation. And then somebody says, right, let's see, could we use that for atopic derm or vitiligo? That repurposing and leading to an approval. So here is Sulzberger 75 years ago thinking the same thoughts. And what he showed was hydrocortisone. It had a fairly moderate effect in atopic dermatitis, and we know that that's because it's a fairly weak topical corticosteroid, but it showed effect, and that was where the era of topical corticosteroids came in, repurposed from rheumatology, really, which is where Kendall was originally targeting his cortisones. What about methotrexate? Well, methotrexate was, again, people mashing up liver. Lucy Willis mashed up liver, got folic acid, and then people got interested into how you would antagonize folic acid. So the structure got fully elucidated in the early 40s, and then people began to think, well, what if you block this? And this was Sidney Farmer and Subaru developed a chemical that would then inhibit folic acid, and they took those through a few refinements from aminopterin and treated kids with leukemia. It was the first kind of targeted chemotherapy at that point at high dose. But then again, people looked at it and said, well, what about folic inhibition? Would that work for inflammatory disease? And of course, we now know it does at much lower doses. So that was then repurposed in psoriasis in the late 50s, and people started using it off-label. It actually did get labelled in 1972 by the FDA after it had been used for about 15 years or so, and it's now used for psoriasis as well, for atopic derm as well. And just that brought me into just a little bit about where drugs get get licensed and approved. So there was really very little formal regulatory approval of drugs in before the 1960s, and this became with the thalidomide disaster really in the late 50s and early 60s. The USA, they had one female scientist who said, we're not going to allow people to take thalidomide, basically because she didn't have a good feeling about it and didn't trust that it was safe in pregnant women. So it never was used in the United States, but it was used in Europe. But the disaster that came from that with the birth defects drove a completely new regulatory framework. And essentially, we got our modern regulatory framework, which is changing a little bit, but it's 2 replicate trials. Drugs have to be safe and they have to be effective. Previously, it was just if they worked, the safety issue was less focused. And we now have in the European Union the EMA that was centralised in around 1995, and that applies obviously to Hungary as well, with different countries bringing it in. So post-approval then, this kind of post-62 approval era, when we began to post-thalidomide era, for example, another example of repurposing, cisretinoic acid, that was designed and failed as a cancer drug for head and neck cancer and for neuroblastoma. Again, somebody in the NHS said, well, why don't we use this for something else? And this is where Accutane or isotretinoin comes from. Somebody seeing it, recognizing the potential, saying this will shrink sebaceous glands, and it's one of the most efficacious drugs that's ever been invented for any condition, but it wasn't invented for acne. It was repurposed into it. Cyclosporin, similar thing, again, discovered as a naturally occurring compound in 1972 for renal transplants. It got approved in the 80s, and then literally 3 years later, it was being used for psoriasis. And this proved that psoriasis was an immunological disease, not a structural disease of the skin turnover, which it had previously been largely considered to be. And then there were double-blind trials for atopic dermatitis very early in France in the early 80s. And this is this is really what was the first study showing a repurposing of a T cell driven drug that then said, look, hold on, psoriasis is actually an immunological disorder, and that opened an entirely new era of therapy. Just to remind you that these drugs, methotrexate and cyclosporin, are still extremely useful. We've much more expensive drugs now available in Hungary as well as all around Europe, but in Ireland and in the UK, there's a requirement to try some of these less expensive drugs first. So there's a trial that Carsten Floor and I did, just a comparison with methotrexate and cyclosporin. Still very efficacious drugs all these years later. And just one thing, is this pointer working? I think, is it? Doesn't point on the screen. Anyway, it doesn't, don't worry about it. But what we're looking at here is this is treatment, and then this is off treatment. So we can see the easy scores, just a severity curve, like the F VASI. This is the easy score coming down, and importantly on the methotrexate arm, there's a disease modification effect for methotrexate, but not so much for cyclosporin. So some of these really old drugs, 70 years plus, are still very useful, still inexpensive, and for a significant proportion of patients will modify their disease off treatment in this section here. So just looking at dermatology as a example, go back to 1972, the era of modern regulation, and you can see it's a almost exponential curve, not quite, of new indications for dermatology. And not only these new approvals, but then of course they get used immediately off-label. So the armamentarium that people have is just extraordinary. In the 30 years I've been doing dermatology, it's really expanded very dramatically. From that's the sort of first 30 years post-regulation to the next 30, it's just there's really no comparison. And then just to take a look at this for immune-mediated inflammatory diseases and where we've gotten to here, we started off with those broad immunomodulators, again, repurposed drugs, methotrexate, azathioprine, a transplant drug that got repurposed, tacrolimus, another transplant drug that got repurposed, made into a topical. Going back to Sulzberger in 1952, the same idea. This works systemically. Would it work topically? But all of these are a little bit more broad. And the question is, could you be more specific? And yes, some 24 years ago, we saw this from Chris Griffiths and others. Chris is also a collaborator of Peter Heggie. We've seen targeted TNF inhibitors working. They were developed for rheumatoid arthritis. Again, people looking at them, repurposing them, putting them into a different condition, and then saying, look, you could have a targeted immunotherapy, immunologically targeted therapy for psoriasis that might work. And this kicked off the era of biologic expansion. So it's just sometimes in serendipitous, inspired repurposing opens up a whole new field that people didn't realize existed before. And it can come from very small series of individual or small numbers of patients that then expands into huge development programs. And then with targeted biologics, we went through adalimumab, and then we went to IL-12, 23. That was ustekinumab, which came in the early noughties, or in the late noughties. And subsequently guselkumab, tildrakizumab, risankizumab, more refined versions, dropping the IL-12 subunit just on IL-23. IL-17 came in then with secukinumab, the first approval 10 years ago, and subsequently ixekizumab, brodalumab, bimekizumab, which is bispecific. I'll come back to it. Anti-IL-4 receptor, which is dupilumab for type 2 inflammation, approved in 2017, and then a couple of other IL-13s, tralokinumab, and more recently lebrikizumab. And then IL-13 receptor, IL-31, which is nemolizumab, approved in Japan and subsequently now available in Europe, or approved but not available yet. At least I don't know if it is in Hungary, but it's approved by the European Medicines Agency, and it may well be available later this year. So you're seeing these very targeted monoclonal antibodies targeting single or dual cytokine pathways that are really transforming a lot of disease. And we've also seen more targeted small molecules, looking at PD4 inhibitors, apremilast. PD4 was a target that was really identified 40 years ago, but subsequently got successfully drugged in 2014 for psoriasis. Doesn't work so well in atopic derm, but topicals like crisaborole, difamilast work in atopic derm. And then topical JAK inhibitors. You've heard a bit about topical Rux, and delgocitinib is another pan-JAK inhibitor that's approved in Japan, now approved for chronic hand eczema in Europe. Again, not widely available just yet, but coming on stream. Baricitinib, abrocitinib, and upadacitinib, all available for atopic derm, in trials for baricitinib is approved for alopecia areata as well as ritlecitinib. So the JAKs have come on, these small molecules making a big difference to the therapeutic armamentarium. You also see TYK2 with deucravacitinib, which it's available for psoriasis. It's not really been quite so successful just because its efficacy measures not so good. And then we have the JAK-TEC family kinase, which is ritlecitinib, approved in 2023 for alopecia areata. Not widely available yet, again, for cost reasons. So again, a simple repurposing and a big expansion of diseases. But of course, there's things that don't work. So if you target epidermal cytokines, and I have a whole list which I won't go through, but the path to success is littered with many failures, and that's something you'll have in your career. Hopefully not many, but you'll have some things that don't work out, ideas that seem brilliant, but really what the evidence didn't show back them up, or things that you tried. And that, of course, that's part of science. It's trying and failing and trying harder again and working out, learning from things that don't work and taking the energy forward into the next. So there's been lots of hundreds of millions of dollars of investment failed in certain targets. So it's not a whole success story. And I would say in atopic derm, which is the disease that I work most closely in the common disease space, people getting to clear or nearly clear skin, still quite low proportions of patients with most agents compared to psoriasis. So we've some ways to go. And for those of you who are dermatologists, it's much further to go again for hidradenitis suppurativa, which is probably the most complex inflammatory skin disease that's most underserved. So what will the future look like for these immune-mediated inflammatory diseases? And I think we're going to see a few things coming through that are really kind of interesting. We've shown the pathways through immunoglobulin therapies, but we're going to see peptides as well. And this is a peptide that targets the IL-23 receptor. It's chemically engineered to just prise open the receptor and not let IL-23 bind to it. And you can see this is the most recent study, the iconic lead study, looking at an oral peptide, icotrokinra, which is, they're always hard to pronounce, these new ones, but icotrokinra, with the retreat and withdrawal arms as well. And just to show you the PASI, so the 90% or 100% of their psoriasis clearing with these drugs. And you can see by week 24, very high proportions of people getting to very deep endpoints on this drug compared to the placebo. And then when you switch at week 16, it really dramatically increases. So we're going to see oral peptides. We're seeing oral molecules coming through for atopic derm as well. Bispecific antibodies are going to be something that we see. So bispecific antibodies have a clone, have a common arm, and then they have two different epitopes, epitope A and B, just nominally, so they can target more than one cytokine at the same time. And there are many, many of these. There's about 20 of these in development. I'll just show you a few of these. These are cytokines that are bispecifics that are in cancer, so they're very prominent in cancer, but not yet in inflammatory skin disease. They're just in early development here. Apart from bimekizumab, which is a kind of bispecific, so it targets two IL-17A and 17F, and that's already approved in HS and in psoriasis. But we're seeing other psoriasis targets like TNF-alpha, which we showed earlier, TNF inhibitors, and IL-17 for more resistant psoriasis. That's in there, in development already. We're going to see bispecific antibodies in atopic derm. I'm only showing an example of about a dozen in each. So TSLP plus IL-13. Most of these have IL-13 as the dominant cytokine plus others. Some are trispecifics coming through in very early stage development. So we're going to see multiple specific targets coming through in single molecules. We're also going to see co-formulation, which is a little bit different from bispecific. So bispecific is a single molecule with two epitopes, or if it's three. But these are co-formulation, usually monoclonals, but you formulate them together, and you can adjust up and down the formulation depending on the week of dosing. So you can go in early with IL-13 for atopic derm, get early control, and then maybe target something like OX40, which is a T regulator like a target, and that might then put people into long-term remission. So these kind of co-formulations becoming a little bit more like chemotherapy regimens, where there's a cycle 1, 3, 4 going right through to cycle 12 or whatever. We're going to see those developed, and hopefully putting more people into either long-term control or long-term remission. And the definitions of those are under active kind of debate at the minute. But we're going to see these in asthma. We're going to see them in atopic derm. We're also going to see nanobodies. So nanobodies come from camelids, usually alpacas, and what they do is lack a heavy chain, so they're much cheaper to manufacture. Camelids have different antibody structures than we do. So they're cheaper to manufacture, and they're also smaller. They're like 10 times smaller. So they're 15 kilodaltons rather than 150. So they have tissue penetration that can get deeper, and therefore you don't need to dose at the same levels of dosing. So you can have lower dosing, better tissue penetration, and cheaper manufacturing in theory. So nanobodies are in trials. They're in late phase 3 trials now, particularly the MoonLake one, sonelokimab in HS. That was their phase 2 about a year and a half ago at the EADV. The phase 3 will read out later this year, and that might be the first nanobody that we've seen in dermatology. It's also in trials for psoriatic arthritis, and the early data look promising there. Protein degraders are a new way of targeting signaling pathways. Probably the most interesting and relevant one is STAT6, but IRAK4 is also in clinical trials. So this is just essentially a medicinal chemistry to dissolve a signaling kinase, usually that's intracellular. So you've got the monoclonals, which will be extracellular, free cytokines, or engaging a target receptor. These are intracellular, and they dissolve the intracellular signaling mechanisms and take them down really quite effectively, more effectively in some cases than a classical small molecule inhibitor that reversibly binds onto something. So we're going to see protein degraders. This is the IRAK4 degrader, which is in just a phase 1 in AD and HS. There's a STAT6, which is currently we've only seen it in healthy humans so far, but it took down that read out just a few weeks ago, and it took down type 2 biomarkers even in healthy humans. So it looks a very promising molecule. STAT6, a company called Chimera, is in development. So that might become like a dupilumab, oral version of dupilumab. So there's a lot of exciting things coming through with these new techniques that can be used. What about small inhibitory RNAs? Well, yes, they're already first in human just in March of this year with Alice Pharmaceuticals. This is a siRNA that takes down JAK1. So it's a way of inhibiting the RNA that makes signaling pathways and taking that down. We're going to see if that works. And that's a topical, it's an injectable. So you're going to inject that. So that's for some existing, that's how we've gotten to where we are and where we might go in IMIDs. But what about rare disease, the other thing that I do? Well, again, we'll go back to repurposing and then drug development programs. So this kind of disease-centric approach, it looks for conditions that there's no treatments for, and then you think, well, is there an agent out there? Look, there's 3,500 approved agents. Could any of those be repurposed into these rare diseases, which are very expensive to develop drugs for? And there's just a few examples that we've done here. This is chronic mucocutaneous candidiasis. It's a STAT1 gain of function. There's also an AIR gene that's recessive, but we're talking about the STAT1 gene here. And this is somebody who's got severe alopecia areata. We know that interferon gamma is a big downstream signal or downstream effector of JAK-STAT. And we thought, well, look, if we give JAK inhibition, we might fix that. And so we gave ruxolitinib. This is like over 10 years ago. She just completely regrew her hair. She'd had autoimmune infertility. She eventually went on and had some children off ruxolitinib. And that was really the first patient to have an alopecia areata recovery on a JAK inhibitor. Now there's 3 approved JAK inhibitors for alopecia areata in people who do not have this genetic background, just people with regular regular alopecia areata. So these are examples of ultra rare diseases, repurposing a drug that came from bone marrow cancer, and then subsequently, it works here, and then a huge development program that I wasn't involved in with hundreds of millions of pharma, and then we have 3 approved drugs. That's kind of how it comes through. That was our original paper back in 2015. This is another guy who's got an ultra-rare condition as well. He's got a mutation in desmoplakin. He's got psoriasis. He's got heart problems. He's got an epidermal differentiation defect. He's got a mutation in desmoplakin. We know that people who have this also have significant cardiomyopathy and a high risk of death in their first year of life. And it's called EKC syndrome. We know that in him and one other patient in Chicago, they're very type 17, type 23 skewed inflammation. So we repurposed, in this case, ustekinumab. You heard about ustekinumab earlier, IL-12, 23. And this is the little Chicago girl on the left and our patient on the right. And they got to clear or nearly clear skin on the ustekinumab and didn't have to attend hospital with multiple infections. Both subsequently had heart transplants. So repurposing the ichthyoses, it works for some 12, 17 and 23s, doesn't work so well in others. And there are many other examples like dupilumab for Netherton syndrome, JAK inhibitors for Darrier disease or Hailey-Hailey, lichen planus, lots of repurposing successes out there. What about vascular anomalies, which is another thing that we do quite a bit of? These are not my patients. These are from Guillaume Canot in the Necker Hospital in Paris. And this is PIK3 kinase related overgrowth syndrome, or PROS. So you can see lots of different features of it, where people have like, hemimegacephaly, polymicrogyria, vascular birthmarks. This would be sometimes called Klippel-Trenaunay syndrome with geographic birthmarks. You might have macrodactyly isolated. So it's a very variable condition. It's mosaic, it's post-somatic. So it depends on the percentage of your genes that have the activating mutation and when it occurred in your development. But it's a very debilitating disease. And this then leads us to think, well, look, if you have an activated PIK3 kinase mutation that's post-zygotic, a somatic mutation, could you target it with a repurposed drug? In this case, alpelisib, which already enjoys an approval for breast cancer. And the answer is you can. And this is Guillaume's paper from 2018, showing really sometimes life-changing effects on repurposing this drug. The FDA subsequently approved it 3 years ago. And we've had many of our patients who are in this approval submission showing their benefits on the drug. The EMA have yet to approve it. What about KRAS gain of function? Yes, so KRAS is another cancer target, and this is again another effect of looking at vascular malformations, where you have the before and after sequence, really dramatic differences in repurposing these drugs into patients with vascular anomalies. And I just want to wind up a little bit on cell and gene-based therapies, which are coming through for ultra-rare conditions. So this is topical gene therapy. You kind of combine phase 1 and 2 trial. You don't have to go through all the traditional phases in ultra-rare, ultra-severe diseases. The regulators will look at that a little bit differently. So this is Peter Marinkovic's data here. It's looking at a wild-type HSV. We know herpes simplex virus targets the skin very effectively. So they have basically a carriage platform of it, and you can put in any therapeutic effector you want. In this case, collagen 7 for recessive dystrophic EB, and that's been spun out by Peter into a company called Crystal, and it's applied locally to the skin. So these are the baseline 3 months for a bunch of patients, probably some of the ones who had the better responders, but it's a really horrible, horrible condition, RDEB. Those of you, it must be just one of the worst things possible to live with or to be born with. And those of you who look after or provide care for these patients will know it's just a really awful disease. So we're seeing here the recombinant collagen 7 through a HSV-led vector improving things. This is what it's called. It's virtually unpronounceable. VGVEC is probably the way to just call it. But it's about 300,000 a year. It comes back to the expense. It's been approved in the EU and approved in the FDA. What about cell therapy? So cell therapy has been around for a long time, maybe 20 years, but it's incredibly difficult to make it commercially or successful or to get an approval. But Michele De Luca from Italy did this study more than 20 years ago and subsequently followed people up. So this is where you take a patient's own stem cells, genetically correct them ex vivo, and then grow them up and transplant them back. And this is, again, the collagen 7A1 using the expansion. So this is now the commercially available one. This is using a murine leukemia virus vector. And then you take the drug and you grow it up and apply it to the individual as a once-off to one area lifetime treatment. Not got approved again, phase 1/1/2A with, in this case, Zevaskin, we're going to call it. It just got approved a few weeks ago, and the ticket price for this is $3 million for a lifetime treatment. So this is where you get into ultra-rare diseases, and the societal benefit and the health economics become really kind of interesting and challenging. Just to talk a little bit about dermatology and economics, these are the top drugs by sales in 2024. And if you just go around, at the top, you'll see Sanofi, Regeneron, Dupixent. That's a big dermatology drug. We don't prescribe the next one, but Stelara is a big one. Mounjaro, people are beginning to prescribe that for both in psoriasis and HS, because obesity is a major comorbidity and driver of it. Skyrizi is obviously risankizumab, widely prescribed. And getting back around to Ozempic, that's quite widely prescribed in some places by dermatologists. Now, I don't, but others do. So you can see where your dermatology is really becoming a kind of a challenge for healthcare budgeters. Not your doxycycline, that's going to be okay. They're going to be happy with that, but this is a bit of a different challenge. So just some conclusions. Like, I hope I've given you a little bit of a tour of to show that, you know, dermatology can be about genetics, it can be about immunology, it can be about pharmacology and drug development, it can be about health economics. There's so many things you can express your interest, skills, talent, and energy in. We have an astonishing array of therapeutic options, and we're going to have more and more, and we'd have to figure out how best to use them is going to be a challenge. Clinical trials are expensive and high risk. You see a lot of failures, but the pipeline's strong. We're going to see a lot new mechanisms of action. And for really, in the first time in the last 5 years, drugs have been tried for the first time in dermatology patients rather than being tried in RA or somewhere else. And that's because dermatology is actually quite easy to get readouts. So you can visually score the efficacy endpoints and get approvals quite quickly. We're still going to be repurposing these 3,500 or more drugs, and the costs are going to be really significant and a major societal challenge where we need all of you evidence-based PhDs to tell us how to use them. So I'm going to wrap up now and take any questions.
No, it's time to ask questions. So this is almost philosophical. I mean, we have more and more very efficient drugs, which are more and more expensive.
Yeah.
So what to do? How the future would?
Yeah, so it's a huge challenge, and the truth of it is, the uncomfortable truth of it is, a lot of these drugs are paid for by the insurance plans of people living in the United States, and the rest of us, to some degree, benefit from that. That's an uncomfortable truth for Europeans, Ireland, Hungarians, everyone else. On the positive side, there's a big patent cliff coming. So patent cliff means you have your patent and you fall off the cliff, you no longer have a patent, and then you get biosimilars if it's a biologic, or you get generic drugs if it's a small molecule that can be easily manufactured. So in the next 3 or 4 years, we will have many more generics for some of the drugs I mentioned, and many more biosimilars, which will allow low and middle income countries access to these drugs, and it will make the health budget more manageable for middle and higher income countries. So I think the patent cliff is a positive. It's inevitable that countries are going to spend more and more of their budget on health. People are getting older, and the options to treat them are better and more expensive. So it becomes really a political public policy thing, where our job is if you're a trainee evidence assessor, you know, to work out what are the benefits people get for this, and as a prescriber to prescribe the least expensive option that gets the job done, so we get more people get access to it. And because anything you spend on this is money you can't spend on building houses in Hungary or Ireland, or giving people unemployment benefit or child benefit. So it's a societal issue and a political issue, but it's not a terrible problem to have. You know, it's better to have these new possibilities and then figure out a way how you make it work in whichever environment that you're in to get to maximize it. But it's better to have the options than not have the options. I think that's why I'm an optimist. Yeah.
Yeah, but as you said, I mean, we still need to know more about the mechanisms of action, because if we know the mechanisms of action, we just don't inhibit in general the immune system, but we can target it actually with small molecules, and that could actually bring the cost down. I mean, questions? Fundy. Fundy she's the one who graduated first from translational medicine, 2 years ago, and actually she already had. Not only, I mean, she serves not only as an SMS, but also as a supervisor. I mean, she showed up actually with one of the fellows. So
on all of the methodological advisor and everything. I saw it, yeah.
Yeah. Thank you, Gabor. Yeah, so my question is like a clinical question that regarding severe atopic dermatitis patients, what is the course of the therapeutic approach in Ireland for you? Because you mentioned that it can be influenced, of course, by the healthcare insurance, and you cannot apply the best drugs for the first time or the first line, for example. And it's the same in Hungary, and I'm just curious that how is it?
So in Ireland, I think it's quite similar. We don't have for those drugs, it doesn't matter if you have health insurance or not, which is, so it's very equal in the sense of if you attend people privately or you come through the public clinics, the same criteria apply and the government pays for it because they're outpatient or community-based drugs. But in order to get, we currently have four advanced therapies that are approved. We have upadacitinib, abrocitinib, dupilumab, and tralokinumab, and probably lebrikizumab is coming on. So in order to prescribe any one of those four drugs, you have to have had severe AD, you have to have tried at least 12 weeks of methotrexate or cyclosporine, and you have to have either not responded or have had an adverse effect. And then we apply on an individual basis with there's an identifier number. I'm sure you have one in Hungary. It's the same idea. It's like a public services number or national, and then that gets individually approved. So that's how our system works. I think it's broadly similar, isn't it? Yeah.
Yeah, so it's quite similar, and I was just wondering if you start usually with cyclosporine or methotrexate.
Oh yeah, so what do we do first?
Just come across patients on cyclosporine, for example, here at our clinic for the first line young patients, for example.
So we use both. If people are not really erythrodermic and really in extreme pain, I'll usually use methotrexate because it works really very well, and it can reset the disease in some ways. And most people tolerate it. We use subcutaneous methotrexate a lot. And some people don't come off it after 12 weeks. They're so happy. They're just like, it's fine. I'll stay on it. Others don't. We only have to try one of the two. You don't have to have failed both. In Scotland, you have to have failed at least two. So the cyclosporine I'll use in people who are very severe, who need quick turnaround. We use it in pregnancy, obviously. You can't use methotrexate, but you can use cyclosporine. So most of the pregnant women I look after who have severe AD are on cyclosporine. So we do use it. I use it in people who've got more nummular eczema, where they've got type 17 elements as well as type 2 elements, that kind of psoriasiform eczema overlap. Cyclosporine is pretty good in that circumstances. So you do use both patient level, and comorbidity. So younger people do better in cyclosporine, they're less likely to get hypertension and so on.
Okay, thank you.
More questions, please.
Hello, thank you for your wonderful presentation. It was very interesting. So I have a question regarding the treatment in HS. So HS is one of the very challenging diseases to treat. And what do you think about the future of HS treatments? Are we getting closer to more personalized treatment, or?
I think we are. I think we've got secukinumab approved, and so is bimekizumab approved. So they're both on the 17 pathway. The issue for HS, I believe, is that we don't know when to start the advanced biologics, and I think we're not starting them early enough in many patients. And the difficulty there is to persuade a regulator that you should start when people have early stage 1, 2, and you're worried about them, because a lot of early stage 1 or 2 respond quite well to lesser age. But we're starting people in the trials who've already got anatomical, tunnels and deep nodules and deep scarring, and it's very hard to reverse that without surgical intervention. So where you start the treatment, I think, is really going to be the most interesting thing. And I think if you start people on the 17s early, their outlook is really very good. when you start people later, it's really hard to know where we're going to go with drugs for that. Like we've seen IL-1s in clinical trials. We've seen JAK inhibitors in clinical trials, and I think their effect size on the high score is all going to be relatively modest. I don't think you're going to see, like PASI 100s or EASI 75s. They're all going to be quite modest because of the patient populations that are going into those trials. So I think we'll still be talking about the best way to manage HS in 10 years. But the important thing is get them on something early before the damage, I think. Yeah, what do you think?
Yeah, I think I agree. Unfortunately, actually, we see mostly that they don't get the treatment in the early stage, and they're not approved for the early stage. And actually, we are planning to do a study on that.
You got to move upstream a bit. That makes the difference.
Thank you.
So thank you for your presentation. I had even 2 questions, both related to generalized pustular psoriasis. My first question would be, in Ireland, in your clinic, how do you manage generalized pustular psoriasis? Because it's very hard as far as I see. And maybe do you have any experience with these novel interleukin-36 inhibitors?
Yeah, no, it's a great question. It is really difficult to manage. We still use cyclosporin and supportive care. The Boehringer Ingelheim who have spesolizumab just couldn't get through our health economics, so they've withdrawn their application. So if I want to get spesolizumab and somebody's in the intensive care unit, I could probably get it, if they were really sick, but I haven't needed it yet because the hospital has a sort of separate, like our budget systems are crazy. Yours, I'm sure, are crazy too. But if you're in the hospital could buy it in as a one-off, but the community approval of it won't be approved in Ireland. So we're not going to have spesolizumab, IL-36 antagonist. But so most of our patients are in intensive care. They're in a exudry, kind of like burns kind of management, and they're on cyclosporin and supportive treatment, antibiotics or other supportive treatment as they need, pain relief. And it's a horrible, horrible thing, and it's not easy to treat. So we don't have anything much better. We sometimes will also use an IL-23, so it could be a cyclosporin plus guselkumab or something like that. So most of my patients will end up on something like gauze, when they leave, but they'll have cyclosporin in the acute. Like, what do you guys do here? Do you have?
We apply the same
Similar?
as well.
Yeah.
Yes. So this is also completely the same in Hungary.
Yeah. So I haven't needed to request it, but I think I might be able to get it for somebody in intensive care, but I don't think I'd be able to get it in any other circumstances. Yeah.
Thank you.
Thank you. So for the sake of time, we have to finish this, although you can continue during lunch break, which is coming. But before that, a very final question. Who is this guy?
He's probably off the internet, I think. He's given his consent, I believe.
Okay.
Thank you very much.
Thank you.